This lecture is part of my lecture series in basic molecular biology. In this lecture, I will overview gene expression, especially with reference to the prokaryotic gene expression, the operon concept. LAG and TRP operons are the two examples. And then I will compare the gene expression in prokaryotes with that of the eukaryotic cells. Gene expression has two levels. One is transcription, another one is the translation. Major control of gene expression is at transcription level, especially the synthesis of messenger RNA, which carries the message. Proteins are the functional molecules which perform the function which is present in the DNA and translated from DNA through RNA. DNA of an individual is same in all types of cells. It is the expression of the genes that makes a difference. For example, insulin gene is present in all types of cells but is expressed only in beta cells of pancreas. The activity of insulin, when it is required, the gene is expressed. So if the insulin gene is expressed in other types of cells, that will be a useless activity. So the expression of the gene has to be tightly regulated, and as I mentioned earlier, that transcription is the major control, although there is some sort of regulation also takes place at the translation level, but that is a minor one. Major is at the transcription. There are different levels of gene expression both in prokaryotic cells and eukaryotic cells. In this lecture, I will focus mainly on operons and especially LAG and TRP operons in prokaryotic cells. But there are other levels such as sigma factor and some other levels that I will discuss in some other lecture. Similarly, I will overview eukaryotic gene expression a little bit, but there is another lecture which is on eukaryotic gene expression with more details. So what are operons? Operon basically set of genes which are expressed under control of single promoter. And you might remember that the promoter in case of prokaryotes, it has two main regions like -10. This is the consensus sequence for -10 and -35 region. And then there is an upstream element which may or may not be present. And this is the place where transcription begins. So I have some basic lecture in transcription in which I have detailed discussion on promoters and other regulatory sequences which you may take if you want. And these operons, these are present in prokaryotic cells. So we go first with the operon. Operon has three genes: galactoside permease, which is shown by lacY, beta galactosidase, which is shown by lacZ, and galactoside transacetylase, lacA. These are the three genes which are controlled by this promoter. And then there is an operator also present here. And then there are some other regulatory sequences which are far upstream. So operon was the first operon that was discovered, and it is well characterized now and well studied. The most studied and well characterized one is the operon. And the function of these genes, basically this operon is for the catabolism of lactose. So lactose cannot cross the bacterial cell membrane. It requires a certain protein which is galactoside permease, which is present in the cell membrane. And this lactose is transported inside the cell with the help of this protein. Once inside the cell, this is the structure of lactose. There is beta galactosidase enzyme that converts this lactose into galactose and glucose. And these enter into the glycolysis for energy. There is a minor product made by beta galactosidase, which is an isomer of lactose. It is called as allolactose, and you can see there is a difference of linkage. Here there is 1,4 linkage, so this is 1 and 4, and here it is 1,6 linkage. So this is the difference between these two. Now, as I mentioned in my previous slide, there are 3 genes which are regulated under one promoter. 2 of these genes are shown here, and these are the products of these 2 genes. This is for the product of lacY, and this is product of lacZ. And these 2 proteins are mainly required for this lactose catabolism. The third protein, galactoside transacetylase, its function, especially in the lactose catabolism, is not very clear. So we will focus on these 2 proteins and their genes in this case. So once again, these are the 3 proteins and their genes which are transcribed under one promoter under operon. And as I mentioned earlier, there's these 2 proteins, they are required for the catabolism of lactose, and all 3 genes are transcribed together producing one messenger RNA, which is polycistornic, from a single promoter, and each cistron has its own ribosome binding site, start and stop codon. It means these 3 genes are transcribed into single messenger RNA, which is translated into 3 different proteins. And you might remember from my lectures on transcription and translation that this process of transcription and translation is coupled in case of prokaryotes. Before transcription is over, ribosome binds to the messenger RNA and starts translation. Now, the very important point for control of gene expression in operon is that the requirement by the cell. So cell first prefers to use simplest sugar, like glucose is used first. When this glucose is depleted from the cell and lactose is present outside the medium, lactose comes inside the cell and then lactose is utilized. It means unless this glucose is there is no need of lactose utilization, hence no use of operon. This we will discuss how these cells sense the presence of glucose and lactose to turn on and off the operon system. So operon is controlled at 2 levels. One is the negative control, another is the positive control. Negative control is like brake of a car. So there is a repressor which is bound to the system, which represses the operon system, and unless you remove that repressor, the system cannot be turned on. Another one is the positive control. It's like an accelerator pedal of a car. It's an activator basically, and this is an additional positive factor response to low glucose by stimulating transcription of the operon. It means this system basically senses the presence or absence of the glucose, and we will discuss in detail. So first, negative control of operon is dependent on another gene which is present far upstream of the operon genes, and this is lacI gene. lacI gene, there are 4 identical polypeptides, so it's for its function. So this is a tetramer of 4 identical peptides, or polypeptides, and it binds the operator. Operator is a sequence which is very near or overlap with the promoter sequence. So this repressor, it binds to the operator and the whole of the operon is repressed. So when it is bound to the operator, RNA polymerase cannot bind at this place, hence the transcription is repressed. And operator and promoter sequences are contiguous, and this repressor bound to operator prevents RNA polymerase, as I mentioned. As long as no lactose is available, operon is repressed. with the help of lacI protein. And how this is induced? So there is a certain molecule which is called as inducer of the operon. So repressor is basically an allosteric protein. It means binding of one molecule to the protein make a change in its conformation at another site, a remote site on that protein. So altering its interaction with second molecule, the inducer binds the repressor and it causes the repressor to change conformation that favors release of the operator. It means the inducer, when it binds to the this repressor protein, due to change in conformation, that protein can no longer bind at the operator region and is released from the operator region, or it cannot bind to the operator region when this inducer binds to that particular repressor protein, which is lacI protein. So inducer, as I mentioned earlier, that there is a minor molecule which is allolactose. It has beta 1,6 linkage. It is an isomer of the lactose. So that acts as an inducer of operon, and this is lactose converted into 1,6-allolactose with beta galactosidase, the same enzyme which converts lactose into glucose and galactose. So this is a minor product, but it has a major contribution that it acts as an inducer of this operon. And in this picture, it is very clear that this is a far upstream lacI gene making messenger RNA, making protein, lacI protein, and 4 of these identical polypeptides, they bind to the operator region, and RNA polymerase cannot initiate transcription from this site. So there is no transcription in the of the operon. But as soon as inducer comes, like 1,6-allolactose, it binds to this protein, and when it binds to the protein, this protein can no longer bind to the operator region. Hence, this place is clear for RNA polymerase to bind and start transcription. And as you can see, single messenger RNA making 3 different proteins. This is the sequence of operator and the promoter region, and this operator, you might notice that it is O1. In fact, there are 3 sequences, O1, O2 and O3, which are required to control this operon. But once again, because this is a basic lecture, I'm not going into much detail of all these operators. I have another lecture in which I have discussed in detail about these 3 promoters and what are their functions and how do they interact with each other. For experimental purposes, this operon can also be induced by an artificial inducer, which is IPTG, isopropyl beta D thiogalactoside, which is very similar to this its inducer 1,6-allolactose. So it functions just like 1,6-allolactose, but this is an artificial inducer and it is used many experiments such as blue-white screening, etc. But that's why I wanted to show you this structure of this artificial inducer. So if I summarize the negative control of operon, it is repressed with the help of a lacI protein, which is a product of lacI gene far upstream from the promoter of the operon. And that protein binds at the operator region, and when inducer comes, it does not allow the binding of that protein at the operator. Hence, transcription can be initiated. So the positive control is basically with the sense of the glucose molecule. There is another protein which is called as CRP or CAP. This is the same protein with 2 different names, cyclic AMP receptor protein or catabolite activator protein. So this protein is an activator, as its name implies, like catabolite activator protein. And cyclic AMP receptor means this protein has affinity for molecules, very common famous molecule cyclic AMP, which is acts as a second messenger in both in prokaryotes and eukaryotes. And the person Sutherland, he got Nobel Prize on the discovery of this cyclic AMP due to his uh, huge importance. So in this case, the presence or absence of glucose is also sensed by lac operon with the help of this cyclic AMP. So in case if this concentration of glucose inside the cell is high, this cyclic AMP level is low. So obviously when cyclic AMP level drops down, it cannot bind to its protein CRP. So this protein is required for activation of this lac operon as opposed to the other protein lacI. LacI protein, which when binds to this operator, it represses the transcription. But the CRP, when binds here, it activates the transcription. It activates the binding of RNA polymerase. Hence, transcription goes on. So this protein can only bind at this place, which is next to the promoter region, when this protein is bound with cyclic AMP. So when would this protein bind to the cyclic AMP? When glucose concentration in the cell is low and there is more cyclic AMP. So in that case, this CRP cyclic AMP complex forms. And when this complex forms, this will bind here as it is present here. So it cannot bind by itself. It can only bind when it is attached with the cyclic AMP or when cyclic AMP comes to this protein. And once this complex binds here, transcription is initiated, provided that lactose is present in the medium and lacI is not bound at this place. Here once again, this is the CRP, this is cyclic AMP, this is the CRP side, this is the promoter side, this is the lac repressor. Now both things are given simultaneously. So there are two conditions for system to become active, meaning lac operon to become active. The first condition is there should be low glucose and high cyclic AMP so that activator protein binds here. And second is high lactose. Why it is required? Because this will be converted into 1,6-allolactose. And this allolactose will take off this lac repressor from the operator region. So it means this lac repressor should not bind at this place and this CRP cyclic AMP should bind. Only then this transcription can take place. Under high glucose, even lactose concentration is high and lac repressor is not bound there, transcription cannot take place because transcription and translation are expensive processes. So it will be useless to make those enzymes when glucose is already present inside the cell. So, or when there is no lactose, even then this system should be stopped working. And same is happening here. So it means system can only go in the positive way in the transcription mode when glucose concentration is low and this protein is bound and lactose concentration is high, is present in the medium, and this lac repressor is not bound at that place. Now, one important question arises that in the beginning, if there is none of these enzymes is there, how can this lactose come inside the cell if these proteins are not there? And if lactose cannot come inside the cell, that wouldn't be converted into allolactose, which is inducer of the system. It means lactose should come in the cell by using galactoside permease, and it should be converted into allolactose with the help of beta galactosidase. From where these proteins come in the first place? Answer to this important question is that there is a basal level of expression always going on, meaning very low level of expression, and this expression is due to half-life of the proteins. When this lac repressor is bound to the operator region, there is a half-life of just a few seconds, and this protein will be degraded. So before new protein comes here, this protein is degraded and new protein has to come to the operator region. So before this takes place, RNA polymerase binds and do the transcription, and translation takes place simultaneously, and it means that few molecules of all three proteins will be synthesized in this condition. When there is no lactose even is present and no allolactose is there, so repressor itself is degraded, and this process is continuously going on in the cell. So what would happen? These proteins will be synthesized. Meanwhile, lac repressor would bound again at the operator region and system will be stopped again. So this process is a continuous process, and those proteins which are made like galactoside permease and beta galactosidase and even transacetylase, all these proteins are degraded and new are synthesized then. So this is a continuous process which is going on unless there is a lactose present in the medium and glucose inside the cell, its concentration is decreased. So in that case, those few molecules of galactoside permease will drag the lactose inside the cell, and those few molecules of beta galactosidase, those will degrade it into glucose plus galactose for further utilization. And now the anabolic operon is the one example is the TRP or tryptophan operon in the bacterial cells. There are 5 genes which are regulated under one promoter and regulatory region, and these are TRP E, D, C, B, and A. And these are the proteins which are synthesized from these genes. There is single messenger RNA having start-stop codons and multiple ribosomes attached here to make these proteins. And you can see tryptophan is ultimately synthesized by following this procedure. But we are not much interested in this lecture, in this part. We are more interested in the regulation of gene expression. So here, similar to the lacI repressor, there is a TRP repressor which is synthesized from far upstream region, upstream of the promoter of this TRP operon. And this repressor, this is messenger RNA and this is protein. This repressor is controlled by the level of tryptophan in the cell. If tryptophan level in the cell is already high, there are 2 places where this tryptophan can bind. And when tryptophan binds to these places, this repressor binds at the operator region and transcription is repressed. So it means high tryptophan level inside the cell controls or stops further synthesis of tryptophan by this mechanism. But the problem here is that this repression is leaky, meaning it's not very efficient. So there is second level of repression or control that also goes on, and that is with the help of this part, the leader sequence. And you can see this is also transcribed, but later on it depends on certain conditions, that if the tryptophan concentration in the cell is already high, this further transcription is stopped. But if tryptophan levels are low, the transcription continuously goes on for the synthesis of these proteins, synthesis of messenger RNA followed by these proteins to make the tryptophan. So how does this control take place? I will give you in the next slides. This is the leader's peptide sequence, and this is the messenger RNA sequence, and this is the site of transcription attenuation as shown here. This is the sequence for that, and this is the first of the polypeptide of the regulated genes, TRPE. This is the sequence for that. So it means leader peptide can also be transcribed and translated to a certain level, but ultimately regulated genes have to be transcribed and synthesized. How this is controlled? Again, there are two possibilities: when tryptophan level in the cell is high, and the other situation is when the tryptophan level in the cell is low. So you might remember the coupling of transcription and translation in prokaryotic cells. This is DNA and this is messenger RNA being synthesized by RNA polymerase. But before this RNA polymerase completes its function, translation is initiated by attachment of ribosome, because there is no nucleus and whole things, both the processes are going simultaneously. There are codons which are present in the messenger RNA. Transfer RNA along with the particular amino acid come to these codons. And other important thing is the sequences 1, 2, 3 and 4. Among these, there is a partial sequence complementarity between 2 and 3, and there is a good sequence complementarity between 3 and 4 sequence. Now there are two possibilities, whether this 2 and 3 they complement with each other, or 3 and 4 they complement with each other. This complementation basically controls the whole transcriptional process in this operon. So coming back to these two codons, in case of high tryptophan level, this ribosome is going very fast on these sequences of messenger RNA, and nothing is there to stop this ribosome. And as soon as it comes to this these two codons, transfer RNA will come right away because tryptophan is there. So before this 3 is synthesized by RNA polymerase, ribosome reaches or covers this sequence 2. What it means? When ribosome covers this sequence 2 before even 3 is it emerges out, this 2 is not available for 3. So because it is covered by this ribosome. Meanwhile, sequence 4 comes out. So it means 3 and 4 will make this intra-chain hydrogen bonding due to this complementarity nature, and this sort of hairpin structure is formed. And this hairpin structure is required in case of prokaryotes for termination of transcription. So same thing will happen over here. Transcription will be terminated, RNA polymerase will be detached, and the regulated genes, the transcription of those genes won't take place. So in high tryptophan level, there will not be any further synthesis of tryptophan due to this structure. But in case at low tryptophan level, what happens is that this ribosome is waiting for transfer RNA along with tryptophan and is paused at this place for some time. Because of low level of tryptophan, the transfer RNA finds it hard to find the tryptophan. Hence, this ribosome is going slow or is even paused at these two codons. So this 2 now is available to make partial complementarity with the 3. So once this partial complementarity is made, when this 4 comes out, 3 is not available for 4. So in this case, 3 and 4 do not make this intrachain hydrogen bonding. Rather, 2 and 3, they make this this type of structure, which is not a hairpin structure. So in this situation, what happens? There won't be any termination of transcription, and RNA polymerase will continuously it will go on transcribing, and the genes, the regulated genes, all 5 genes, TRP, A, B, C, D, and E. These are the structures. This is the hairpin structure. When 3 and 4 they pair with each other, and this is the structure when 2 and 3. This is the partial complementarity. So this structure cannot terminate the transcription, but this structure terminates the transcription, and difference between these two makes this regulation possible at the TRP operon. This is another example of gene expression in prokaryotes where multiple genes are controlled by a single repressor, like lacI repressor. This is not an example of operon, but multiple individual genes, they are repressed by a repressor which is lacI repressor. Under severe stress conditions, SOS response takes place, signal of stress. For example, if E. coli cells are continuously under UV radiation, they will try to escape from as much as possible. So, and they will try to repair the damage. So this lacI repressor is bound basically to the operator regions of the genes which are required to repair the damage, any type of damage. Because these genes are needed at once, there is another protein which is REC-A protein. That protein is already present, but its concentration is high under stress condition. And this protein also contains a coprotease activity. So coprotease activity helps in degradation of many different proteins, including lacI repressor. So once this lacI repressor is degraded, all these genes start expressing. There are many other examples that are available in literature, but these were the most common one which I wanted to discuss in this lecture. Now coming to the eukaryotic gene expression, there are certain differences between these two which I will highlight. First is that in prokaryotes, RNA polymerase generally has access to every promoter, and it can bind and initiate transcription at some level of efficiency, and it does not require any activators or repressors, meaning transcriptional ground state is non-restrictive. Whereas in eukaryotes, you know, there are certain histones and certain other proteins, they bind to the DNA molecule, So strong promoters are generally inactive in the absence of regulatory proteins. It means transcriptional ground state is restrictive unless some sort of activator comes in the picture. These two conditions are given in general situation. There may be specific examples where these two situations may not imply, but generally this is the situation in prokaryotes and eukaryotes. This is overall hierarchy of eukaryotic transcription. Eukaryotic messenger RNA is synthesized by RNA polymerase 2 along with its many factors bind over there. In promoter region, there is a Tata box which is recognized by Tata binding protein, and RNA polymerase 2 is attached with this protein. But whole of this can only be possible when there are certain other proteins come in the picture, like transcription activators bind to the enhancer elements. Enhancers are, these are the sequences present in the DNA where certain sort of activators bind. They have the DNA binding domain and the protein binding domains. And then there are modification and remodeling enzymes which actually modify the chromatin structure, and remodeling enzymes help in the to give the access through the RNA polymerase and other proteins so that they can bind to the DNA molecule. And then there are certain HMG, high mobility proteins. These proteins also help in bringing these activators closer to the promoter regions. And then there are certain proteins which are collectively called as mediators. They have the function that they on one end interact with the activators, and on the other end they interact with the RNA polymerase 2. So whole of this these factor interaction of these factors is required in case of eukaryotic transcription. So this is overall picture. It will be more clear from here. I go one by one. Like there is a enhancer element or sequence in the DNA which is recognized by the activator protein. One part of the protein attaches here, other part of the protein is available for attachment to the modification and remodeling enzymes and the mediator. So when these things they come over here, then comes certain transcription factors that recognize Tata binding protein also recognizes this Tata sequence. And finally, other transcription factors and this RNA polymerase 2 that they come in the picture. So along with mediator and all that, and transcription can be initiated after phosphorylation of the CTD. So from here on, I have discussed in my lecture in transcription. But it means this transcription is initiated by binding of these activator proteins. Now the question is, what are those signals which instigate these proteins to bind to this sequence for initiation of transcription? There are basically two main types. One are hormones, steroid-like hormones or other molecules, or thyroid hormones, retinoids, or vitamin D regulatory part. So those type of hormones which are steroid-like, they can cross the membrane because of their hydrophobic nature. and they travel in the blood with the help of certain proteins. When they reach to their specific site, these hormones are released. They can cross the membrane, go inside the nucleus, and inside the nucleus there are certain receptors. These hormones bind to receptors, and these then are attached to this region. So they are basically now they are the activator proteins, so which bind to the hormone response elements, and further reactions or further interactions take place, as I mentioned uh, just previously, that how these modification and remodeling enzymes and mediators and RNA polymerase 2 comes in the picture. And here one example of the hormone which cannot cross the membrane, such as insulin. It binds, it has receptors on the membrane. Because of hydrophilic nature, it itself cannot come inside the cell. So when it binds to its receptors, inside the cell there is a change take place. Like here, phosphorylation takes place, and then there is a cascade of reaction. Different proteins, they come in the picture. They are phosphorylated or dephosphorylated, and like a relay race, you can say. And finally, one of those can go inside the nucleus, where it acts as an activator protein. So it instigates the mediator and other remodeling modification enzymes, and finally the RNA polymerase to start transcription. Uh, in another example, some molecules, they can cross the membrane. Even these are hydrophilic, like glucose, but they require certain transport protein for going inside the cell. And once inside the cell, further transformations take place. Like in this case, it is converted into xylulose 5-phosphate, which helps in the dephosphorylation, partial dephosphorylation of carbohydrate response binding protein. When this protein is fully phosphorylated, it cannot go inside the nucleus. But when half of it is dephosphorylated, it goes inside the nucleus. And once inside the nucleus, it is further dephosphorylated. And now, along with some other factor, it can bind or act as an activator to interact along with mediator and other proteins to start the transcription. And this picture shows the complexity of eukaryotic gene expression. This is phosphoenol pyruvate carboxykinase promoter region, and you can see how many different proteins are there to initiate this transcription with the help of RNA polymerase 2. So overall, you could imagine that the gene expression in case of eukaryotes is quite complicated as compared to the prokaryotic cells. So with this, I end my lecture on overview of gene expression. Thank you very much.